Before we have Dr. Zager come up and talk about the Delcast study, we want to turn it over to have just an open forum to ask questions of our doctors. So we'll do this for about 10 to 15 minutes. So feel free to ask any questions you have. And pop up your microphone so we can all hear you.
I'm a Catholic class 2. 2 years, 6 months postdoctoral. 2 years, 3 months post-class.
Can you turn your microphone? If you push that button, it'll pop up.
And evidently there was not enough tissue other than projecting, enough to do anything else with. But I've been led to believe where I thought class 2, it really didn't matter when you radiated the tumor, that didn't affect the survivability percentage. And as I understand what you said earlier this morning, you see it may affect the percentage. And earlier to be treated better.
It's possible, and this is still very early, and we're not talking about days or weeks or even months, but it could mean that that if we find a class 2 tumor when it's 1.5 millimeters thick and 5 millimeters in diameter, and it looks just like a nevus, and we treat it then, which is earlier than anybody's treating them now, that it might, that could have an impact. But if it was treated in, if it was detected in June and it was treated in December, that doesn't make a difference. That's not what we're talking about. We're talking about probably years earlier. But that's where the problem lies, is that when they're that small, they overlap a lot with nevi. So, there's some that are arguing that we should just be treating a whole bunch of nevi in order to hit the few that really need to be treated, but then we're going to be treating a lot of people that don't need treatment. So, yes, there's a possibility that that at the very early stages, if we can treat them earlier at that point, that it could make a difference.
You were talking earlier about that one and some of the other things. Is there any other way to find out whether or not you have those after the fact?
After you've had treatment, you mean?
Right.
There's yes, the short answer is that
it depends on if you had a biopsy at baseline, and if it was, if extra tissue was preserved. Like in our center, if patients are part of our study, we always save extra tissue, not tissue, the sample, the DNA sample. So that would matter. It can be checked after treatment if it's not too long. If you biopsy several years after radiation, let's say, it's mostly going to be just scar tissue left. There's not going to be much to really biopsy. But what our hope is that we'll develop new blood markers that would also allow us to detect these in the blood. But that's a few years away.
Bill, that's actually news to me. So if I plat somebody 6 months ago and then they would like to, actually we biopsy over 90% of our patients, I think, for the gene testing that Castle offers. I think it's maybe 100%, something like very close to 99%. But let's say someone moved into Colorado and they had been plaqued 6 months ago, and they're showing a nice response, the tumor's shrunk a little bit, and they would like a biopsy. Is that something I can do and get some useful information out of?
Well, we're working towards that. The answer is, I think the answer is yes to that. And now we think that the gene expression profile would probably change in 6 months, and we know that chromosomal gains and losses will change after radiation. But if all we're looking for is a BAP1 mutation, we might still be able to find that. I think we will still be able to find it. But it does need to be validated. It hasn't been validated for clinical use, and that's part of what we will be looking at very soon.
The reason I sound a little surprised, and he explained that, the nuance is this: the radiation will damage the DNA pretty badly. So the GEP test is done on kind of looking at a global expression of genes. And I would assume that would be radically altered after radiation has shot your DNA to pieces. However, that is kind of a random thing. If you're looking at one gene, the BAP1, that makes sense.
Yep. So that's definitely something we're working toward. You know, it didn't make sense to focus too much on BAP1 for prognostic purposes when the best tests out there from MIT and MD Anderson, and we're missing 20% of them. But in my lab, we've developed new techniques where we're missing less than 5% of them now. So we can actually move towards using BAP1 as a prognostic marker, BAP1 mutations as a prognostic marker based on these new findings. And that's part of what we'll be doing in the COOG-2 study, is validating this.
If you don't mind, I'm going to ask you. Oh, yeah, go ahead. This is about you guys. Then.
5% or 20% of what population?
So we think that virtually all class 2 tumors have a BAP1 mutation, but we can only find it using the existing publicly available technology in 80%. So that means 80% of the time you can find the mutation, but in 20% you can't. So you don't want to use that alone to replace class 1, class 2, because 20% of the time you're going to be telling the patient the wrong answer, at least. But we're getting better and better at finding, the problem is that BAP1 mutations can be, it's like finding one misspelled word in War and Peace, okay? Or maybe, maybe an A becomes an E, or something, like something very hard to see. So we're using very sophisticated supercomputer technology to find all of these mutations. But as that becomes more commonplace, and we can find 95 or 98% of the BAP1 mutations, then that could be very useful as a prognostic test, as well as a test used to decide which therapy to use. Okay. And that could be used in these radiated tumors, because that mutation is not going to change after radiation. Your chromosomes are going to change, your gene expression profile is going to change after radiation, but the mutation, if it's there, it's still going to be there, so you should be able to find it. Yes?
So I had my surgery done at University of Colorado, and good consent for them to be able to use that for research. Is that research shared with other entities, or does that stay just at that university?
I don't know. I'm not familiar with what Dr. Oliver's doing there. I imagine they're probably doing a little bit of their own stuff. The beautiful thing that Dr. Harbour's test has done is it's united all of us with the common testing. And prior to that, there was, I call it provincial testing. Every little academic center had to have their own little test, right? That's how academics can work. But I think there's still that going on. There's still independent investigation going on in parallel to the Castle Biosciences test. And I think that's likely what they're doing over there.
Is there a way that you can release that information to other?
Oftentimes research, you sign a consent, and part of that consent is that they'll de-identify you. So when they do get results, they may not be able to backtrack it to you. So I'd encourage you to talk to Dr. Oliver or those folks over there, get more information about what that is exactly.
Yeah, I'd be happy to talk to you offline about, because every IRB is different and they all have to kind of talk together, and there's this whole other level of regulatory compliance that we have, but
The HIPAA laws are federal.
right, would be happy to talk to you about that
Serious
offline.
laws, yeah.
Yes, sir.
Back to plaque therapy for a second. Why iodine versus ruthenium or palladium? What's the case for that?
Well, we could use palladium. The difference between palladium and iodine are really the half-life. Half-life meaning how quickly does it decay away. So they're essentially the same energy. We have the same type seeds. So the difference between palladium and iodine is that palladium will deliver its dose much quicker. So it becomes a radiobiological effect, because while all this is happening, the cells are repairing themselves, they're maybe dividing again. So that's one thing that a clinical trial would help us answer. Is there a benefit for using something like palladium that will deliver it faster than iodine?
Most of us think that there's not a big difference between the local control or, I mean, what we're balancing is local control versus visual outcomes. And some radiation sources are very powerful, but they fall off very quickly. So you can get a very high dose locally, but a millimeter or two away, you're not getting much. That works great. That's like ruthenium. But ruthenium falls off very quickly, so if the tumor's more than a few millimeters thick, you won't be able to kill the tumor. So you've traded off local control for better vision, which is, you don't want to do that. Iodine and palladium, in the old days there's something called cobalt that people used, which was very powerful, but it caused a lot of radiation damage. So iodine and palladium are kind of a balance, you know, in a non-technical physicist language, a balance where you're trying to balance those two issues of getting the tumor completely treated while not having a lot of extra scatter of radiation going around. My opinion is that there's not a big difference between palladium and iodine in terms of their local control or visual outcomes. They're both good. The question I have is, what role could ruthenium potentially play? And the history of ruthenium is really interesting. The only source for ruthenium plaques traditionally was in East Germany. So because the United States was at war with communism, we couldn't even get ruthenium plaques because they were made in a communist country. So generations, did you know this?
No.
Yeah, generations of ocular oncologists in the United States were trained without any ruthenium because of that, right? So a few of our colleagues have trained in Europe where they did use ruthenium, and they came back over here wanting to use ruthenium. The ruthenium is potentially useful for very thin tumors that are close to the macula or the optic nerve, where you're trying to limit damage to those structures. It's still hard to get ruthenium in the United States. And in Canada as well. It's hard to get it. There's a lot of regulatory issues, and you know, anytime a center starts using something that it hasn't used before, there's a learning curve too. So it hasn't really caught on over here. I think that it would be nice to have it. I'd probably use it, I would guess, in 10 to 15% of cases where I now use iodine. So it's not going to make a big difference, but it could make a difference in a few selected cases. But between palladium and iodine, I personally don't think there's a big difference. Do you feel differently, Peter?
I don't think so. I liked what you said. There's sort of a marketing aspect. Sometimes I feel people hear that competitive marketing. You may hear somebody say that that's more advantageous, preserving vision, that kind of thing. But no, and I think it's, we've looked into that, and my understanding is the cost hasn't worked out. It was going to be very expensive to make that happen for whatever situations we've looked at.
And the thing is, once you have something that has 30, 40, 50-year history like iodine, it's been through a 20-year, you know, NIH study, and something else, and people have a lot of experience with, the physicists know it well, we know it well. Something claims to be 2% better, it's hard to change the whole system over 2 or 3% change that may or may not be real.
I want to follow on that. That's an excellent thought. I mean, the CMS study was a very large, expensive, beautifully run trial, and we got a lot of very useful information out of that. Our radiation physicists and our radiation oncologists feel like if we do everything the same way and follow essentially the CMS protocol, then we have an expectation on outcome. You have what I call an orphan disease, really. You know, it's rare. We're not going to be able to fine-tune treatments. You know, breast cancer, a lot more common, there's a lot more things going on. Here, if we stick to what we have, we know it works pretty well, and we know that people should fit in to an expected outcome. But that was, again, 10 years ago. It finished, and times have changed, and there's newer technologies, and so it's a little nerve-wracking when you go onto a clinical trial, or if you deviate from a protocol as a physician. And I like to use the term, I'm like a river raft guide. We're going down the river. What am I supposed to do to help you if I'm not really sure what to expect with something new? It's kind of hard on me to make a recommendation. And yet, this is the way science is done. We have to go forward, and we have to take those steps cautiously, carefully, usually within a well-constructed clinical trial. So when people are doing things that are not really gone under rigorous trials, it's hard to know what you're talking about there because it hasn't had that scientific proof behind it, and it falls into what we call anecdotal evidence. Again, you guys being an orphan disease, if you're less than 4,000 cases per year incidence in this country, I think they consider it orphan, if you will. You've all heard that term. I guess it's the poor orphan that nobody else is looking out for you, right? And it's tough. So I, again, want to compliment you guys for coming together. This is a really good group you've got. I know there's a lot of other people that participate that are not at this meeting. But by doing this, the thought is you can leverage research. And I'm glad to say that last year, there's a very competitive grants that are given out by the OMF. And this year, it was awarded to Dr. Harbour's lab for just an outstanding proposal. So I'm getting off track here a little bit, but I think there's a lot of power into what you're doing. And these questions are great, and I would encourage you to stay involved.
So we actually need to stop the questions now.
He's been asking several times.
Okay, one last question, but these guys will have office hours, so they'll be out here and you can ask direct questions of them.
You believe, based on your research, has a 90% chance of having the BAP1 melanoma?
Yes, at least 90%.
Can't prove it, but that's...
Well, we can prove now in unpublished data that it's over 90%, so at least 95, but we don't know if it's 100%.
That occurrence of the BAP1 may actually give some clues to therapies.
That's right.
Therapies to treat.
That's correct.
Would it then hypothetically behoove the person who's class 2 and doesn't know whether they have the BAP1 get another sample taken from their tumor even a couple years later, assuming the BAP1 would still be there, to ensure, they've got the BAP1, therefore this therapy can be.
I think I see where you're going. You're asking Dr. Harbour, but I think that may be the case that we may be talking about that down the road, Bill. If you're looking at refining the class 2, looking at the survivors that have class 2.
Well, I think what we were talking about before is the person who did not get prognostic testing. They come in 6 months later, they would like to have prognostic testing. I think your best bet is to check for the mutation for the prognostic purposes rather than a chromosome test or gene expression profile, which will both change after radiation. So that's a prognostic test. But the question you're asking is, if I already know I'm class 2, would it be of value to go back in and look for BAP1? I think the answer is no, because the likelihood is so high that there's a BAP1 mutation. I don't think you need to put yourself through that. I think you should just assume that there's a BAP1 mutation.
Would a physician hypothetically actually treat someone as if they had BAP1?
Many do, yes.
What was the question?
Well, yeah, go ahead.
Would a doctor actually prescribe a therapy for somebody, not knowing for sure they had BAP2, but knowing for sure they are class 2 or BAP1?
Yeah, the data we published were that about 85% of class 2s had a BAP1 mutation. That's 6-year-old data. Now we can say that over 95% have a BAP1. So, and we're finding new ones every day. We're looking at other people's published data where they said there was no BAP1 mutation, and we're finding the BAP1 mutation. But we're having to develop innovative new technologies to find the BAP1 mutation. So that's going to approach 100%. It may not reach 100, but it's going to approach 100%. So I think it's reasonable if you already know your class 2 and to not go back and rebiopsy to know if you have a BAP1 mutation.
Oh, fantastic.